Welcome back, students. Welcome to the fifth video of our online classes in pharmaceutical microbiology. In the last class, we have seen various culture medias and how they were prepared, what were the composition, and how various culture medias are used to culture various types of microorganisms, right? And you should know once the culture media is prepared, what is it used? It is used to grow the bacteria, right? So how do they grow the bacteria? So first they'll be taking a solution of a bacteria or the solution having the bacteria or bacterial cells, then that will be added to the culture media, and then they will put it for incubation in order to grow that. So there are various techniques of isolating the bacteria after their growth in the culture media. So in today's class, we'll be studying about the isolation of pure cultures and bacterial growth curves and how the bacteria are increasing in the culture media. So today's class objectives will be, the first thing, to know the basic types of culture. There are basically two types, mixed and pure. We'll see what they are, and after that you need to learn the methods of culture techniques. In what way are the uh, microorganisms inoculated? In what way are the microorganisms added to the culture media in order to grow them? And after that, we'll go on with a bacterial division, how they are dividing, and then we'll go with the phases of bacterial growth by looking at a graph, the bacterial growth curve. Okay. Beginning with the basic types of culture, there, as I just said, there are two types of culture. First is mixed culture, and the next is pure culture. What is mixed culture? Mixed culture contains different types of bacteria within them. Uh, when a culture media is taken and a mixed culture is inoculated into the culture media and incubated, after 48 hours, we'll be able to see different colonies. You can see the image here. This is the petri dish into which the solid media, that is agar media, is present. Okay, and in that you can see different colors. One is red, one is orange, one is whitish, one is yellow. That means different colors are forming, which means different colonies of bacteria, which also means different bacteria, right? So in the mixed culture, it will be a mixture of all bacteria, not all. It will be a mixture of more than two bacteria. Okay, and whereas pure culture, it contains only one type of bacteria, and we will be able to see only one type of color when cultured. You can see the image here. This is the petri dish with agar, and this yellow color is just one type of bacteria having same color, that is yellow. Okay, this was types of culture. Now moving on to the methods to isolate pure cultures. There are three methods, basically: streak plate method, pour plate method, and spread plate method. These are the common techniques that are followed in order to inoculate bacteria, inoculate microorganisms into the culture media, incubate them, and identify them, what type of bacteria they are. So beginning with the streak plate technique. In this technique, what are the basic requirements is a needle or a platinum rod or a glass rod. Platinum rod, a glass rod, or a needle, or you can say platinum needle also. Okay, that platinum needle will be heated so that it turns to red hot, and then cooled for 30 seconds. Okay, that will be a sterilization technique. When we are heating the rod, all the bacteria present, that will definitely die. And another thing is, we need a petri plate and a solid medium in that. So basically, petri plate will be taken and a melted agar will be added and allowed to cool. Okay, and after some time, the petri plate will be solidified. You can see the image here. This is solidified petri plate. Again, the type of medium, you can see brown color, that is heated blood medium. Okay, this will the petri plate. Okay, they will first heat the needle or the inoculating rod and dip into the solution having bacteria. So they'll take a sample. It can be either a soil. They'll take a small amount of soil and add it in the water and mix properly, and that will be a solution of a bacteria or bacterial suspension, because soil also contains bacteria, right? So the soil solution can be used as a bacterial solution, and the rod will be dipped into the bacterial solution, taken out, and streaks will be drawn. You can see the images here. Initial inoculum, Streaks will be drawn first in this way, and the next one in this way, and next in this way, and this way. So there are four different streaks or four different corners of the streaks. During this process, it should be the person should be very much careful that he do not destroy the solidified medium. It should not be disturbed. It shouldn't be disturbed even a small bit, because if it is disturbed, the bacteria will go below the nutrient medium, and it may not grow there. So it is must that the one who is inoculating the culture media should be very careful. And after inoculating, they have to incubate it. They can incubate either for 24 hours and 48 hours. It depends on the type of microorganism which we are culturing. After 48 hours, we'll be able to see this type of petri plate. So this is the streaks drawn in the beginning. Okay, it can be this way or in the zigzag way, this way, two types. And after incubating, either 24 hours or 48 hours, we'll be able to see these type of colors. Okay, here, and there's no here, which means the first stick that we draw is will have a good concentration. The second stick that we draw will have a little bit less. Third will have more less, and fourth will have more less. So it makes us to the first stick will be a dense line. You can see here it will be completely dense because it is a higher concentration. Then the next streaks are a little bit lower. That means a diluted one, and this one is more lower, and the last one is further more lower. And by the last streak, we can easily identify which type of bacteria there. And in case of this one, we keep on putting a streak in zigzag manner. So in the beginning, it will be very much denser, and as it moves on, it will be very much smaller. Okay, and this way we can identify which type of bacteria. And here we can also be able to isolate them separately. Okay, here the colonies are different, a bit far. There is a difference between the distance of two colonies. So based on that, we can also isolate them. This was a stick peg technique. In pour plate technique, basically the nutrient medium is mixed with the bacterial suspension, and it is diluted in a serial wise, and that will be added into the petri plate and incubated. So let us see what it is. So first the broth culture, the broth nutrient medium having the bacterial culture is added, mixed, and 1 ml of this is added to another test tube, and 9 ml of saline is added. Then 1 ml of this one is added to another test tube, 9 ml of the saline is added. Again 1 ml of the solution is added to this test tube, and 9 ml of saline is added. By this way, you can see 10 power minus 1, 10 power minus 2, 10 power minus 3. That means the concentration is keeping on decreasing. Okay, then this will be added to the petri plate. Okay, first this one and this one and this one in different petri plates, and this will be incubated. So after pouring the mix with circulator motion, okay, as we add, we need to just circulate so that it is uniformly distributed. Okay, after that, we'll incubate into the incubator. It could be 24 or 48 hours, and after that, we'll be able to see in this one. Plate 1, which is 10 power minus 1 concentration, has more number of colonies. 10 power minus 2 has a little bit less, and 10 power minus 3 is furthermore less. By this method, we can easily isolate the pure culture. So you can see if we have to isolate from this, it is completely dense. There will be no space to isolate a particular bacteria or particular colony of a bacteria. Whereas even in the second concentration, it could be more number of the colonies. Whereas in third concentration, it will be very less, and it will be easy to isolate them. So there are certain disadvantages of pour plate technique, and they are: microorganisms are trapped beneath the surface of medium when it solidifies. So as we are adding bacterial suspension into the broth solution, it is completely mixed, right? And when we are the mixture, when we are adding to the petri plate, it is possible for the bacteria to settle on the bottom. Okay, as a result, the bacterial culture will be from the top to the bottom. Okay, it will be completely like a nail impregnated into the wall. Similarly, the microorganism will be impregnated into the solid medium. So it will be difficult to isolate a particular part. In order to remove, we have to completely scrap that area and then proceed with purifying it. That will be a difficult task, right? So the first point in disadvantage is that microorganisms are entrapped beneath the surface of the medium and solidified. So very difficult to isolate and count the subsurface colonies. And next is it is tedious and time consuming and requires skill. Okay. And the microorganisms are subjected to hot shock because liquid medium maintained at 45 degrees centigrade. What happens is there are certain bacteria which survives in cold environment, like that of cyclophils, below 10 degrees centigrade. Such bacteria, if it is added to the Newton medium, which is at 45 degrees centigrade, and added to the petri plate, what happens? Because of high temperature, that will be destroyed, right? So this method is not suitable for such kind of bacteria. So I just said this method is unsuitable for isolation of psychophilic bacteria. So this was pour plate technique. Moving on, spread plate technique. This is the best method and simple method used in order to isolate bacteria. So what is done is 0.1 ml of the solution is prepared. The bacterial solution is first prepared. 0.1 ml of the bacterial solution is prepared, and then 1 ml of it. Okay, 0.1 ml of the bacterial solution is poured onto the solid medium, that is on the petri plate. Okay, this is bacterial solution and this is the petri plate with Newton medium. It could be agar. Then after a small 0.1 ml is added, we take a needle, bent needle or a platinum rod or a glass rod and start spreading it, first beginning with the upper and lower side. Okay, we spread such that it is uniformly distributed on the solid medium. And after some time, when we incubate it, we will be able to see the colonies here. And the best part of this method is all the microorganisms which are forming the colonies are only on the surface. They are not below the surface of the Newton medium. So this was the best advantage. So we'll see the advantages now. It is a simple method, and in this method only surface colonies are formed, not below the surface. And microorganisms are not exposed to high temperature, as we are first adding this Newton medium on that, and then allowing it to cool so that the Newton medium gets solidified. Then we'll be adding the bacterial culture. As a result, bacteria won't be exposed to higher temperature. So this was the advantage of spread plate technique, and we have discussed first was streak plate, then pour plate, and spread plate. Now moving on to the bacterial growth curve. In order to study bacterial growth curve, we first need to understand three concepts, and they are first there are certain concepts to learn. What is bacterial generation, and uh, how do we tell the bacteria growing, if it is in size or in the number of cells? That will be seeing certain terms, and how bacteria is dividing basically by binary fission. Okay, and what are the phases of bacteria? Moving on with the concept of bacterial generation. Growth. What do we understand by growth? If we consider humans or plants, when we say the child is growing or a plant is growing, it means the size of a plant is growing or the size of an human is growing, right? But this is not the case in bacteria. All the bacteria are of same size, or the same type of bacteria, same size. They won't increase in the size. So in this, the meaning of growth means it is a growth of number of cells, not the size. Okay, growth and increase in number of cells, not increase in size. And bacterial species only maintain if population continues to grow. Okay, bacterial species, if bacteria is present on Newton medium, it will survive only if the bacteria is growing, because some bacteria will die and other will take birth. I mean, it will divide and form a newer generation, right? And this way, bacteria continuously grow, then only it can be maintained. And generation, what is meant by generation? How the bacteria is forming? It is by basically binary fission. We'll see how binary fission is. Then growth rate. What is meant by growth rate? Whenever we use the term rate, it means it is there will be a denominator of time, a particular instant of time. What is the rate of growth? So if I tell 10 microorganisms have been grown in 10 seconds, so it will be the rate of growth will be 10 microorganisms per second. If it is 10 seconds, 10 microorganisms per 10 seconds. So that will be a growth rate. How is it counted? number of cell number by time or cell mass by time. Okay, and next is generation time. The generation time means the time taken for a microorganism or the bacteria to double its size. If suppose if 50 bacterial cells are present, the time taken for this 50 bacterial cells to convert to 100, that is the generation time. So let us see how binary fission actually happens. You can see the structure here. This is the bacterial cell and this is the circular DNA, and circular DNA is called as plasmid, right? So this DNA undergoes replication to give another strand of DNA, that is double helix form now, double stranded DNA. Then it elongates for division, that is the beginning process of cell division. And then the double stranded DNA, circular DNA starts separating and forms in this way septum formation, and then two cells will be separated, and then we form a complete new cell with two different bodies. So same bacteria having two different bodies, that is, it is dividing from one cell, it is forming two, from two it can form four. So there is an exponential growth. 2 4 4 4 into 4 is 16, 16 into 16 is 64, in that way. So there are certain bacteria which are given in the table, and the generation time as well. So Escherichia coli, E. coli, it takes 17 minutes for it to get doubled. So Bacillus megaterium, and it takes 25 minutes. And this is the media used. And Streptococcus lactis, it takes 26 minutes. Streptococcus lactis of the lactose broth takes 48 minutes. Okay, and let us see this one, lactobacillus acidophilus, which is in the curd, right? If that, we have seen this method that our parents add small amount of the curd in a milk and put it in the night, and by morning it will turn to a curd. So why is it happening? Because lactobacillus acidophilus from the curd that we are adding into the milk, that keeps on dividing, right? That keeps on dividing. It takes around 66 to 87 minutes for it to completely divide. If you had 10 cells, it would be around 10, it keeps on exponentially increasing, right? So if you take a small amount, it doesn't have 10 cells, it could have millions of cells, because they're very tiny and they can accumulate very large in number in smaller space. Okay, and Rhizobium japonicum, it takes 344 to 461 minutes. Mycobacterium tuberculosis, in synthetic way can it doubles in 792 to 932 minutes. Okay, and Trypanosoma pallidum takes 1980 minutes. That is, it takes lots of hours. Moving on with just phases of bacterial growth. So how bacteria is growing? Let us see the graph to it. There are four different parts of the graph. First is lag phase. Second is log phase or logarithmic phase. Third is stationary phase, and the fourth is death phase. Let us see what they are. You can see the graph here. The first, this one, 2, 3, 4. There are four different stages, so four different lines. So in this stage, the bacteria growth will not be seen. The bacteria, whatever is added, that will increase in size. How will it increase in size? By consuming a nutrient medium that is present in the culture. We are adding the bacteria to the culture. That means it starts consuming those nutrients in order to grow in size, and then they get divided. So first will be for certain period of time, it will be a straight line. That means the bacteria are just consuming the nutrients. Okay. The next phase is the log phase, or the logarithmic phase. In logarithmic phase, whatever bacteria has consumed the nutrient medium, all those bacteria starts dividing at one time. Okay, that division is rapid. That for that reason it is growing exponentially. If it was a slower process, the growth will be like this. The growth will be like this, but it is not so. It is just like this. So there is an exponential growth, and in the next slide we'll see how it is exponentially growing. And the third stage is stationary phase. In this stage, the growth of bacterial cell is equal to the death of bacterial cell. In this case, in logarithmic phase, only the growth is taking place. There is no death of microorganisms that is happening. Only the growth is happening. For that reason, it has shown a peak to the upwards. In the stationary phase, some of the bacteria will be growing, and simultaneously other bacteria will be dying, right? So for that reason, the bacterial growth will be equal to the death of the bacteria. For that reason, this uh, straight line, you can see plateau in number of living bacterial cells, rate of cell division is equal, and death equal, roughly equal. So rate of cell division is equal to the rate of death of the bacteria. And the fourth stage is the decline phase, where uh, there's no nutrient medium in the first stage, that is lag phase. It has consumed the nutrients and it has divided. The stationary phase, due to less nutrients, some will be dying, some will be dividing. So that will be an equilibrium stage. Whereas in the fourth stage, there are no further nutrients for the microorganisms to consume, right? When there are no nutrients, the microorganism cannot survive. As a result, it keeps on dying. As a result, we get exponentially declining phase. So the fourth phase is also called as decline phase. So this is a lag phase, no increase in number of living cells, but because the bacteria will be consuming the nutrients. Log phase, all the logarithmic phase, in its exponential increase in number of cells takes place. The stationary phase, this is a plateau, that is a straight line, where we'll be saying rate of cell division is equal to rate of death of cells, and decline phase, exponential decrease in number of living bacterial cells. These are the phases of bacterial growth. And as discussed just now, logarithmic phase, there is an exponential increase, right? So this is one bacteria, okay? It one in some time it becomes two, okay? And this will divide to form four, and for this one also divide to form four, and this one again 4, 4, 4, 4. That is 16. Then 16 forms 60. 2 to the 4, 4, okay, 1 gives 2, 2 gives 4, 4 gives 16, 16 gives 32, 32 gives 64. That will be exponential increase. So you can see the generations 1, 2, 3, 4, 5. First is one microorganism or bacteria. In the first generation, there are 2 cells. The second generation, there are 4. Third generation, there are 8. And fourth generation, there are 16. And fifth generation, there are 32. Similarly, exponential value 2 power 1. Okay, 2 power 1 is 2. Then 2 power 2, that is 4. And 2 power 3, 2 power 4, 2 power 5, 2 power 6, 7, 8 keeps on increasing. This is called the exponential phase. And what could be the reason for the bacteria to enter the stationary phase? We have, as just discussed earlier, bacteria will first consume the nutrients in lag phase, then multiply in the log phase. But when does it enter into stationary phase? When the nutrient medium is sufficient for some bacteria to grow, and some bacteria there won't be nutrient sufficient, so they'll die. So the nutrition is limited. Okay, nutrition limitation and limited oxygen availability. Some bacteria will be consuming the oxygen, whereas it will be absent for other organisms, right? So those organisms which cannot consume oxygen will die in case of aerobic, and which consumes oxygen will be surviving. And toxic waste accumulation, as the death of cells are increasing, right? Bacterial cells are increasing, it can accumulate and cause toxicity. And critical population density ratio, as discussed, both the cells are present. One is alive and one is dead. When both are more in number, that will be more in concentration, right? So it means critical population density reached. So in the fourth stage, that is the decline phase, what could be the reasons for that? The first thing is nutrient deprivation. As discussed, nutrients will be consumed in the lag phase, and in stationary phase, limited nutrient will be there, and the fourth stage, there will be no nutrient. As a result, bacteria won't get any nutrition, they just die. So nutrient deprivation, waste material accumulation, many cells are dying, so accumulation will be more. Irreversible loss of the ability to reproduce. As they are dying, they cannot be reproduced, right? And the death can be logarithmic and can vary with environmental conditions. As we have seen in logarithmic phase, they are increasing, and same way in death phase, they are decreasing in a logarithmic or exponential way. Such as the growth medium employed in the it depends on the microorganism to what extent decline phase is and how the graph is. Okay, and there are certain bacteria in decline phase which are resistant, which doesn't die and maintain its integrity, whereas some other bacteria die. So this was the characteristics of death phase, and these are the references. Pelzar, as said earlier, it is the best book to study all the topics, and another is Biotechnology book by David B. class BA. This was today's class. In today's class, we have studied what were different types of techniques and what are different types of culture media, basically, that is pure and mixed. And then we have seen the techniques of culturing or isolating pure culture. There was a stick plate method, four plate method, and spread plate method. And after that, we have seen uh, the growth of bacteria, what are the concepts in bacterial growth, and then we have seen how bacteria is dividing by binary fission, and also we have seen how the growth of bacteria is happening by looking at the growth curve of the bacteria, which has four stages, that is a lag phase, log phase, and stationary phase, and decline phase. Okay, and in the next class we'll be studying about various spectroscopies, not spectroscopy, it is all about the microscopic techniques. And before that, we'll be studying how do we measure the bacterial cell, in what way are they increasing, and how many cells are present, what is the mass of those cells. By calculating one cell, we can also calculate the mass of the cell, right? So if I have one cell and if I find amount around 10 cells, suppose I have one cell and I have seen a lot of the whole colony I've seen. So how will I find the number of cells? So if I take one cell and find the mass of it, then I can check the mass of the complete colony and predict that it can have so and so cells. There are different techniques to identify the growth of cell or measure the number of cells present. That we'll deal in the next video. So thank you for watching the video.